Hello students, how you been doing? Wishing you all the very best for your upcoming exams. This is your faculty Dr. Meenakshi Sundaram A S, and I have been your faculty in StupireMed for both subjects microbiology and biochemistry. Welcome to this particular swift course in microbiology where we try to learn microbiology in under 12 hours. This will be a rapid session. I will not be teaching you things very deeply. I'll teach you the most important things which can help you crack your exams. So these would be the most important things that you have to know before going for your exams. So on that basis, let's begin with general microbiology. Many questions have been raised from the concepts of general microbiology. So remember one thing, general microbiology does not necessarily mean it's a single chapter from which one or two questions can come. Understanding general microbiology will take you forward. Understanding and knowing things in general microbiology can help you understand systemic microbiology better, so that systemic bacteriology, virology, mycology, parasitology, whatever it is, you will try to understand how an organism behaves by looking at this particular concept. So let's begin with the core concepts where we try to understand terminologies first place. In case of microbiology, when you use the word infection, what does it mean? The mere entry of the organism into the host cell is called as infection. Pathogenicity means what? The ability of the organism to cause disease. It is to cause disease. So, you will understand the difference here. All infectious organisms need not be pathogenic. But if an organism has to be pathogenic, it has to be infectious. So just because an organism has entered into the body, there is no guarantee that it will cause disease. But it will require certain pathogenic features for an organism to cause disease. That brings us to the question, what is meant by disease? Suffering, which is obvious. It can be seen as a symptom or can be elicited as signs. It means the organism has entered into your body. Your body is responding to the organism. For example, if a small mosquito is floating around us, and if I do nothing about it, the mosquito can bite me and can leave. But if I want to slap the mosquito, when I make a slap, this slap can attack the mosquito. Imagine the mosquito dies or the mosquito escapes. It doesn't matter. But I have slapped myself. So there will be some kind of redness on my cheeks. So in the process of attacking a microbe, if I suffer, then that is called as disease. So infectious organisms can be pathogenic, need not be pathogenic. But if an organism is pathogenic, it should have caused an infection first. So there can be questions about the routes of entry. For example, there was a question, Legionella is transmitted from person to person, which is not a true statement. Now that brings us to the question, what are the routes of infection? Infection can have multiple routes. An organism can be inhaled. If it is inhaled, it goes through the nasal tract, it goes to the respiratory tract. The ultimate organ can be lung. The organism can be ingested. It can go to the oral cavity, through the food pipe, it can go to the stomach, it can go out to the intestine, it can come out of the body also. Or it can attack any of the part of the body in that particular pathway. It can be inoculated. A needle prick injury where HIV can be spread into another person is because of inoculation. Something from outside is creating a portal. Through the portal, a microbe is capable of entering into the body. That is called as inoculation. Then how does it differ from invasion? No other portal is being created. When the microbe is placed on the skin, the microbe can drill a hole and can enter inside. This is the innate property of the microbe to go deeper. And inoculation means somebody is helping the microbe to go deeper. So if I inject through which the bacteria is going inside or the virus is going inside, it is inoculation. And if I place the virus is capable of penetrating through the skin, it is called as invasion. So these are the four most common routes by which infection can happen. Now, when I asked you about this question about Legionella, right? Legionella is acquired by inhalation. True, very true. But it need not happen from man to man. Why? Because Legionella is acquired in areas like hot water tanks, coolant towers, air conditioners. If people who are working around these areas, they will be inhaling the aerosols throughout the air because the organism is circulating so many times in huge load around these kind of structures. But if a person like a human being is exhaling air, the Legionella does not come out through the exhaled air so easily, so it will be extremely less in load for the opposite person to acquire the organism. So Legionella can be inhaled, but the person-to-person transmission is extremely minimal. Now this is not the same in case of measles. Measles person-to-person transmission can be very high. You don't need an extra human reservoir per se. It is not mandatory per se to actually start infections because of measles. So I hope you understand the difference here. Next, if we look at the concept of virulence. Virulence should be understood at multiple levels. Please remember one thing. All pathogenic organisms need not be highly virulent. So, virulence speak about the ability of the organism to increase the frequency and increase the severity or both of the disease process. Now, it need not necessarily be a disease process. It can increase the frequency and severity of the complications. Now comes a question. What is disease? What is complication? If a human being is responding to an infection, his immune system is trying to attack the organism. In that process, you suffer. That's called as a disease, I told you. So, if I say gonococcus is an organism, and what do I expect from this organism? The disease process is called as gonorrhea. If gonococcus causes gonorrhea, it's a disease. And this is a disease affecting your genitourinary tract. You can have burning micturition, you can have painful micturition, you can have pus coming out in the urine. So pyuria, dysuria, both can be present as a part of your gonorrhea, which can be a type of urethritis. But if the same gonococcus is exhibiting its invasive property, it is piercing through the genitourinary tract and enters into circulation. Through the circulation goes to the brain. In the brain, if it causes meningitis, then that is called as complication. Gonorrhea is a disease, while meningitis is a complication. Why? What the organism can do. If we expect that, and that is exactly happening, that's called as a disease. If the organism is doing more than what is expected, or unexpected dangers are coming out, it's called as a complication. So, gonococcus is known for causing gonorrhea. Gonococcus can cause meningitis, but not known for meningitis. If you look at the opposite example called as Meningococcus. Meningococcus, unlike gonococcus, is not sexually transmitted. It is transmitted by inhalation. The organism travels up the nose, nasal mucosa, attaches to the olfactory bulb. It travels the olfactory tract, pierces through the cribriform plate of ethmoid bone, and enters into your brain to cause meningitis. So here, when it causes meningitis, it's a direct impact. So, for meningococcus, meningitis is a disease. It is not a complication. But for gonococcus, it's a complication. Now, what can be complication for meningococcus? Because meningococcus can become a part of your circulation. It can lead to meningococcemia. It means the meningococcus along with its toxin is spreading through the blood. Now, through the meningococcemia, you can reach your synovial fluid in the joints. In the joints, you can expect arthritis. Possible. So, when meningococcus is traveling through the blood and can reach your joints to cause arthritis, generally arthritis is unexpected from that of meningococcus. This will become a complication of meningococcus. While meningitis is a disease, the same thing can be said for gonococcus. Gonococcus can lead to gonococcemia. And in the blood, it can actually go to the joints to cause arthritis. Though the frequency of arthritis is more in case of gonococcus compared to meningococcus, arthritis is because of systemic circulation and the spread of the organism through the blood. So arthritis is a complication of both gonococcus and meningococcus, while gonococcus will be Causing meningitis is a complication, while meningococcus will cause meningitis as a part of disease. I hope you understand what exactly is the difference between that of complication and disease. So what is virulence? Any factor that helps the organism to establish residence. Look at the most important words here. Establish residence inside the host cell, because of which the organism increases its persistence inside the host cell, because of which it stays inside the human being for long, because of which it increases the frequency or severity or both for the disease and complication, which may lead to death also, is called as a virulence factor. Remember, we don't call a species of an organism to be called as virulent. Whenever I use the word virulent, it is applicable for a strain. All Staphylococcus aureus may be pathogenic, may not be pathogenic. If I pick up a pathogenic Staphylococcus aureus, in that a specific strain is what we can concern ourselves with being called as virulence. So virulence is a word we use for a strain, not for the organism, not for the genus, not for the species. So I hope you understand the difference between these words as terminologies. So with this basic terminology, let us try to understand microbial infections per se. Microbial infections can be made up of 4 major groups of organisms under MBBS curriculum. So we'll be having bacteria, viruses, fungi, parasites. When we focus on bacteria and viruses and fungi and parasites, the first question that we have to raise is which ones are eukaryotes and which are prokaryotes. When I raise such a question, the concept of eukaryotes or prokaryotes fails in case of viruses. Viruses cannot be called as eukaryotes or prokaryotes because some people call them as non-living infectious structures. Some people call them as non-living infectious structures. But most of the people have agreed that it is a living organism. And what kind of organism are they? They are extremely simple kind of living organisms because they contain a capsid called as covering and an internal nucleic acid. Nothing other than that. Just the covering and the internal nucleic acid, be it RNA or DNA. Covering, internal nucleic acid. That's all. Nothing else. But you can decide whether the bacteria, fungi and parasites come under eukaryotes or prokaryotes. Among the three, the only prokaryotes are bacteria. Bacteria are the prokaryotes. So, fungi is a eukaryote. Parasite is also a eukaryote. That brings the question. Parasites can be further differentiated into protozoa and metazoa. Those who are unicellular microscopic organisms are called as protozoa, while multicellular and macroscopic organisms can be metazoa. Not always everything is actually macroscopic. Some can be macroscopic, some can be microscopic. So protozoa and metazoa are the two forms of parasites and these come under subkingdom. If Protista is actually the kingdom, then protozoa and metazoa are called as subkingdom. They all are eukaryotes. So that brings us the question, what is a eukaryote? What is a prokaryote? So let me make it as short as possible. Listen very carefully. Who is a prokaryote? Pro. It speaks about the one who has seen everything. The literal meaning of prokaryotes is something who has seen everything. But in case of this situation, here it means before. Karyotes means the ones who had karyons. And karyons mean nuclei. So, those organisms who emerged and evolved before the evolution of nucleus are called as prokaryotes. It means what? The prokaryotes are those organisms who don't have a nucleus. Instead, they have nucleus-like material called as nucleoid material. Nucleoid material which may contain RNA and DNA. Nucleoid material. Remember one thing, whenever I try to draw a three-dimensional shape of a particular cell, the cell's three dimension will tell you the nucleus is not a circle as we see in the textbook. It's a sphere that we see in the real-time picture. It's a sphere. From the sphere's outer layer called as nuclear membrane, the spaces between the nuclear membrane will go for evaginations, not invagination. They'll go for evaginations, which can start spreading in different directions in 360 degrees, and they can become a part of other kind of organelles. The nuclear extensions are called as organelles. When we have a nucleus, the nuclear membrane will actually start spreading. As it keeps on spreading, it can become other organelles. Because the concept of nucleus itself is a failure for case of prokaryotes, because prokaryotes began evolving before a proper architectural arrangement of nucleus, they will be called as the organisms who don't have a nucleus. So because they don't have nucleus, they won't have any kind of associated organelles. No nucleus, no organelles. It means no mitochondria, no Golgi apparatus, no vesicular ribosomes, no innate other activities. So lysosomes are also absent in case of your prokaryotes. All these are present in case of eukaryotes. So the eukaryotes are the ones who contain a proper nucleus. If, for example, I draw a nucleus here, the nuclear membrane can actually extend like this. This extension can be seen as Golgi apparatus or endoplasmic reticulum. Here, the immediate extension is actually the reticular network fibers. And if I have ribosomes attached to them, then they will be referred to as rough endoplasmic reticulum. From there, the extension can happen further. These extension can give rise to Golgi apparatus. So all these are present in case of eukaryotes and they're absent in case of prokaryotes. Now, there have been certain MCQs raised in case of prokaryotes and the energy requirement. We all know about one thing. Mitochondria is the powerhouse of any cell. But prokaryotes came without mitochondria. So how does a prokaryote survive in the absence of mitochondria? Simple understanding. Listen carefully. Prokaryotes do not need a mitochondria for ATP synthesis. If this is the cell membrane can go for invaginations whenever there is a need for ATP. And the prokaryotic cell will contain the cytochromes which are required for electron transport chain scattered everywhere. Once the invagination happens, all the kind of cytochromes will come and sit on the surface and they will form the electron transport chain. When this electron transport chain happens, the electrons will move from one place to other. At the end of it, oxidation happens, which will be coupled with phosphorylation. ATP will be generated, just as you see in case of eukaryotes. Once ATP is generated, the invaginations will disappear and they'll go back to normal state. So these kind of structures happened only in the intermediate state between the absence of ATP till the presence of ATP. So that is why they are intermediately formed bodies, which are referred to as mesosomes. So you will be having makeshift arrangement of certain structures in the cell which will do the function that can be done regularly by fixed structures in case of mitochondria, eukaryotes. So eukaryote, we have a fixed structure for ATP synthesis called as mitochondria. In eukaryotes, we have a fixed structure meant for keeping your proper packaging of proteins and fixing address on the proteins called as Golgi apparatus. That is also present in case of eukaryotes, whose equivalent can be found in case of prokaryotes in the form of mesosomes. So mesosomes are intermediately formed bodies which will take up the function which is not seen in case of prokaryotes but are regularly done in case of eukaryotes. So that prokaryotes will never be short of any of these functions. ATP synthesis, protein folding, all these things can happen without fixed structures, the structures which are happening to be a makeshift arrangement. Okay, so let me repeat one last time. Mesosomes are the prokaryotic counterparts for eukaryotic ATP synthetic machinery. Let's understand this part here. Okay, so we have realized the basic concepts between that of how bacteria will be behaving as prokaryotes. Now, let's look at a basic structure of a bacteria here. Look at this. Here we have cell membrane, cell wall, capsule, sex pilus, plasmid, attachment pili, flagella, DNA, ribosomes, cytoplasm. Here the DNA is not a nuclear DNA. It's a nucleoid DNA that makes all the difference. Why? Because if it is a nuclear DNA, inside the cell, you will be having a special space occupied by a covered area called as nucleus. The nucleus will have a double-layered nuclear membrane. Between the nuclear membranes, you'll have the nuclear pore. Through the nuclear pore, whatever mRNA synthesized can escape into the cytoplasm. Such things are not existent in case of prokaryotes. That is the beauty. Prokaryotes are existent even without all the kinds of high-sounding structures like these. Okay, now let's try to understand the common things that are present in case of a cell. When we speak about the cell of a gram-positive versus gram-negative bacteria, we have to learn something important. The covering in case of gram-positive and gram-negative are distinctly different. What are the most common questions raised in this area? That is my only target before I start telling you this. See, in case of a gram-positive organism, we have a very thick cell wall. And beneath the thick cell wall, we have a cell membrane. Now, this is in sharp contrast with what we see in case of gram-negative organisms. In negative organisms, we have a cell membrane as the outermost layer, followed by a thin cell wall. Beneath that, you have one more cell membrane. So, when they ask you, among the gram-positive and gram-negative, which one contains a sandwich of layers? The answer is gram-negative. Who's sandwiched? The cell wall is sandwiched between that of the cell membrane called as outer cell membrane, another cell membrane called as inner cell membrane. So, sandwiching is a property of gram-negative organisms in terms of structures and not a property of gram-positive organisms. So, as I told you, there are only two layers in case of cell wall for gram-positive organism, a thick cell wall and a cell membrane. Now, let us come to the question, what is common to both? The cell wall is common to both. So, what is the cell wall made up of? Cell wall is made up of a peptidoglycan meshwork. Peptidoglycan mesh work. What do you mean by peptidoglycan? It's a combination of peptide components with the glycan components. The peptide components are the ones who are proteinaceous in nature, who are extremely antigenic. Who are the ones? We have amino acids called as D-alanine, and we have peptides made up of D-alanine called as D-alanine peptide. We have a special amino acid called as diaminopimelic acid, and we have another amino acid called as D-glutamate. Watch very carefully. All these amino acids are numbered and lettered as D. The D isoforms are useful for structures, especially in case of bacteria. If you try to connect this with that of your biochemical pathways where you come across amino acids, they all will be L amino acids and they are involved in humans and they are parts of metabolic pathways. They are parts of metabolic pathways. Why am I telling you this? Because these will not be a part of metabolic pathways per se. They can't be broken down because they have to be structures. These are supposed to be structures which are covering the bacteria. So all these are the peptide components of the peptidoglycan. Now look at the glycan components. Glycan would be carbohydrates. The carbohydrate contents are the N-acetylated version of glucosamine and again the N-acetylated version of galactosamine. This is one type of the carbohydrate layer. The next type of carbohydrate layer would be N-acetyl muramic acid. Now this is a very common area where people have raised questions. Let me tell you where the questions can be. First, when we speak about D-alanine, this is an important target for the drug called as vancomycin. Vancomycin comes and binds to the the organism's target called as D-alanine. So this vancomycin will inhibit D-ala. And the most common organism who depends on vancomycin to be killed would be your enterococci. Now you have emergence of vancomycin resistant enterococci. These enterococci will not be having D-alanine. Instead, they'll be having D-alanine D-lactate. There is a deamination of the second amino acid called as alanine. Alanine will become pyruvate, and pyruvate on reduction will become lactate. So when it becomes D-ala D-lactate, the target has been altered. Vancomycin will not be able to bind. Because vancomycin can't bind, then the enterococcus has escaped from the attack of vancomycin. So this is an important MCQ. The next thing is you have N-acetylated muramic acid. People have raised questions and asked people about muramic acid is present in which group of organisms. Is it a gram-positive organism or a gram-negative organism? All students will jump together and say gram-positive organism. Do not make the mistake. N-acetyl muramic acid is present in both gram-positive organisms and gram-negative organisms. Why? Let's zoom out again. I spoke about muramic acid as a part of the carbohydrate content of the peptidoglycan meshwork, which is a part of cell wall. And I told you cell wall is thickly present in case of gram-positive organism and still thinly present in case of gram-negative organism also. So the mere mention of the word cell wall indicates cell wall is present in both gram-positive and gram-negative organism. Then the students have asked me, okay sir, if at all you say muramic acid is unique for both gram-positive and gram-negative organism, so it doesn't make it unique, then which structure makes gram-positive organism unique? The answer is an acid called as teichoic acid. Teichoic acid differentiates between that of a gram-positive organism because it is present in gram-positive organism. It is absent in case of gram-negative organism. So teichoic acid is a marker for gram-positive organisms. At the same time, you can ask me, what is that particular present in case of gram-negative organism but not present in gram-positive organism? The answer is aromatic amino acids. Aromatic amino acids are a property of gram-negative organisms, usually not found in case of gram-positive organisms. This is what I want you to remember. So this is about the structures. N-acetylated glucosamine, galactosamine, N-acetylated muramic acid, all these structures are part of cell wall, and cell wall can be a property of both gram-positive and gram-negative organism. That takes us to the next question. Then what are the components of the cell membrane in case of a gram-positive organism or in case of membranes in case of gram-negative organism? Now here something is unique. The inner cell membrane of gram-negative organism will be almost the same as the cell membrane of a gram-positive organism. But what is unique for a gram-negative organism here? The component that is present in a gram-negative organism especially is the outer membrane of the gram-negative organism. This outer membrane of a gram-negative organism is referred to as a lipopolysaccharide. This is a very important source of MCQs. What is lipopolysaccharide also referred to as? The shorter version is anyway LPS. The lipopolysaccharide is technically the endotoxin of the gram-negative organisms. Endotoxin of the gram-negative organisms. Why? Because the lipopolysaccharides is made up of lipids and polysaccharides, obviously. What are the lipids? The lipids are made up of beta hydroxy myristic acid. The lipid component is referred to as lipid A. Beta hydroxymyristic acid, lipid A. Then we have polysaccharides. Polysaccharides means it will be made up of multiple types of sugars, right? So we have multiple polysaccharides which can be made up of glucose, fructose, or variant of any other kind of sugar. That doesn't matter. Why? Because lipid A is the one that gives the uniqueness for the whole lipopolysaccharide. Now, here let me fetch you a question. All lipopolysaccharides are not endotoxins. Listen very carefully. All LPS can be equal to endotoxin. This is a statement. But this statement has an exception. Only when the LPS is made up of lipid A, Only when LPS has lipid A, then the LPS is equal to endotoxin. But if the lipid is made up of anything other than lipid A, for example, I said beta-hydroxy myristic acid. You remember? This beta-hydroxy myristic acid, if it is replaced by any other acid, if I have beta-hydroxy palmitic acid, stearic acid, etc., then it cannot be lipid A. So, only when it is lipid A, the LPS will be amounting to become endotoxin. But if L is not lipid A, then LPS is not equal to endotoxin. So I can frame a statement. All endotoxins are definitely made up of lipopolysaccharides only. There is no question of doubt about it. Because if it is an endotoxin, the toxic property is because of beta-hydroxy myristic acid. So when I say it's an endotoxin, it contains lipopolysaccharide where the lipid is specifically lipid A. But if I mention just lipopolysaccharide, I am not very sure the lipid is made up of lipid A always. So all LPS may not be endotoxin, but all endotoxin can be LPS. This is the statement you can derive from what is given in the textbooks. But there is one small query in this area also. I say all endotoxins can be LPS. That is a statement, right? Some endotoxins can be LOS, which is lipo-oligosaccharide. Lipo-oligosaccharide. Because the lipid A, the beta-hydroxy myristic acid is the content that makes all the difference. If beta-hydroxy myristic acid is deficient and if it is not there, then it is not lipid A, then that lipid containing polysaccharide is not endotoxin, right? So the oligosaccharides or polysaccharides does not actually contribute to the toxic component. So I can make a statement that the toxic property is purely because of beta-hydroxy myristic acid. This contributes to the toxic property. So, even if a lipo-oligosaccharide, it doesn't make a difference. And which organisms depend on lipo-oligosaccharides? The answer would be Neisseria. Neisseria contain LOS instead of LPS, and they can still maintain the same endotoxic properties of other bacteria. Now you can ask a question. Are there any organisms who have lipid A and non-lipid A components? Is there an organism who has no lipid A? Yes, there is an organism who does not have lipid A. It has an altered lipid. It is LPS, but not qualifying to be called as endotoxin. Who is the organism? The organism is Bacteroides fragilis. Bacteroides fragilis is a very common organism. It's the most common anaerobic commensal of the human GIT. If it is the most common anaerobic commensal of the human GIT, it means it is normally present in your body, so it can't be extremely dangerous for the human body. So for that reason, evolution-wise, it is understood that Bacteroides has given out and has sacrificed the property of being endotoxic. So it is not endotoxin anymore. It contains LPS, but the LPS is not equal to endotoxin. Is there any purpose this statement serves you? Yes, because Bacteroides fragilis is the most common anaerobic commensal, but it is the least pathogenic in terms of causing sepsis or DIC. It does not cause sepsis, it does not cause DIC so easily. It is known for causing local peritonitis and abscesses, but it is not toxic like any other kind of gram-negative organism. So the ability of it to produce sepsis and DIC is almost nil. This is the beauty of learning about Bacteroides fragilis. So once again, all LPS need not be endotoxins, but all endotoxins can be LPS, and lipo-oligosaccharides also can contribute to the endotoxic property. But the point is, lipids are the ones who contribute to the toxic proportion, not the carbohydrate. The best example can be Neisseria, who has an LOS, and the LOS is acting as the endotoxin.